Welcome to our lesson on enzymes, which we define as proteins that function as biological catalysts to speed up the rates of chemical reactions without being used up themselves. Remember, in every living organism, there are metabolic reactions that are taking place. So these are the normal reactions that take place in cells, and they are of two different types of the metabolic reactions are of two different types. We have what are known as anabolic reactions, which are those reactions in which molecules are built up. So this involves the joining up of small monomer units to make large molecules. For example, where amino acids are joined up to make proteins, where glucose molecules are joined up to make large molecules like starch. We also have what are known as catabolic reactions. Those reactions that involve the breakdown of large molecules to make small molecules. For instance, starch can be broken down to glucose. Proteins can be broken down to amino acids. So those are the metabolic reactions that are taking place in the cells of living organisms. Can either be anabolic reactions or catabolic reactions. Now, these reactions proceed at very slow rates. So they are too slow such that if they are allowed to proceed at those rates, the rate of production, for example, of glucose would be too slow to sustain life. Remember, glucose is a requirement for the production of energy by respiration. So if we allow those reactions to proceed at their natural rates, they would proceed very slowly. So catalysts are therefore needed. A catalyst is a substance that speeds up the rate of reaction. So catalysts are therefore needed to speed up the rates of these metabolic reactions. So this is where enzymes therefore come into play. They function as biological catalysts, speeding up the rate of chemical reactions without being changed by the reactions themselves. So in the cells of living organisms, we have these proteins known as enzymes, whose role is to speed up the reactions. How do the enzymes do this? In order for any reaction to be initiated, or in order for any reaction to take place, there is some energy barrier that needs to be overcome. That is known as activation energy. So that activation energy is the energy required for a reaction to start happening. And enzymes lower this activation energy. So let's look at this graph, which shows the activation energy required for a particular reaction. As we can see, for us to begin producing these products, this energy barrier or the activation energy has to be met. So what enzymes will do, they will lower the activation energy such that the energy required to initiate this reaction or for this reaction to begin is now much lower than was required before the introduction of the enzymes. So enzymes lower activation energy, and therefore they enable reactions to occur at a fast rate. Now, because enzymes are proteins, they have certain characteristics. We have already stated that they are made of protein. That is their first characteristic. Enzymes are specific in their actions. So that means one type of enzyme will only act on one particular molecule. That molecule on which it acts is known as the substrate molecule. So the molecule acted upon by the enzyme is known as a substrate. So the enzymes will only act on particular substrate molecules. So enzymes are specific. Enzymes are not altered at the end of the reaction. Remember, when we defined enzymes, we did mention that they remain unchanged. So an enzyme will catalyze a reaction, and at the end of the reaction, it releases the product and is able to catalyze the reaction again. In other words, it is able to accept more substrate molecules and continue carrying out the reaction, and it will remain the same, or it will remain unchanged. Enzymes are required in small quantities, and that is simply because enzymes get reused. They are not changed at the end of a reaction, therefore they can be reused. Therefore they are required in small quantities. That is just one and the same point again. They speed up a chemical reaction even at low enzyme concentration, so they are required in small quantities. And enzymes are affected by environmental factors such as temperature, and they are also affected by pH. We are going to discuss how temperature and pH affect enzymes, in our next lesson. Now, looking at the mode of enzyme action, we have stated that enzymes are specific to one particular substrate. So, in this diagram, this is our enzyme. So, these two represent our enzymes. And these enzymes have these specific shapes. We have a shape there, and we have another shape. These specific shapes that are possessed by enzymes are known as the active site. So every enzyme has an active site, and the active site has a particular shape. The substrate molecule that will be catalyzed by the enzyme needs to have a shape that can fit into the active site. So if the shape of the substrate molecule cannot fit into the active site, like in this case, this enzyme will not catalyze the reaction that has this substrate. So because of that, enzymes are specific. So they are specific because the enzyme's active sites have shapes that are complementary to a substrate. If the shape of the active site is not complementary to that of the substrate, then the enzyme will not catalyze the reaction because the substrate will not be able to fit into the active site of the enzyme. So the mode of action of enzymes is often likened to a lock and key. It's usually referred to as the lock and key hypothesis, because only a key with a specific shape that can fit into the lock can be able to unlock this key. In the same manner, like we said, the enzyme and the active site of the enzyme and the substrate have to be of complementary shapes. If we take this key, it wouldn't be able to unlock this particular lock. So enzymes are specific, just like the manner in which keys and locks are specific to each other. This key will only be able to unlock this particular lock. Now, how do enzymes actually catalyze? We're going to look at the steps. First, we have the substrate entering the active site of the enzyme. So this represents our enzyme, and this green structure represents our substrate. So the substrate enters the active site of the enzyme to form what is known as an enzyme substrate complex. While it's still in the enzyme substrate complex, the enzyme breaks the substrate down. So we now have our enzyme, and when our substrate has been broken down, we now have our products. So we now form what is known as an enzyme product complex. Then the products can now leave the active site of the enzyme. Let's look at the shape of the enzyme at the end of the reaction and compare it with the shape at the beginning of the reaction. It's still the same. Remember, enzymes remain unchanged, or they remain the same at the end of a reaction. So that means this same enzyme can then go back and catalyze more substrate molecules, and the same process is repeated until the reaction is over. In our next lesson, we're going to be looking at the effects of temperature and pH on enzyme action. Thank you.